Greetings! In this module, you'll learn how to use the web app Seed Selector, a tool to support seed selection decisions for restoration projects. My name is Kyle Doherty, ecologist, software developer, and author of the app. In this module, we will learn the underlying principles of climate similarity, review app features, conduct an example analysis for a region of interest, and discuss basic and advanced applications of this approach. Climate is a selective pressure in plants and can be a driver of genetic differentiation leading to local adaptation. Thus, matching the origin climate of plant materials to a target restoration site can improve the chances of successful establishment. One tool to guide practitioners through the selection process is the app Seed Selector discussed in this learning module. Seed Selector creates a map of the best climate match given a set of plant materials of known geographic origin, and can also rank the climate match of that set at a particular site. To understand how Seed Selector operates, we must first understand the concepts of climate space and climate similarity. We will examine the state of Utah in the United States as an example. Here we extract two pieces of climate information, mean annual temperature and precipitation for this region, and plot them in the panel on the left. We refer to the resulting cloud of points as the climate space. Each point in climate space has a corresponding geographic location, 1 kilometer by 1 kilometer pixel, in the state of Utah. Let's pick a point in southern Utah, the white dot in the panel on the right, and compare its climate to that of areas throughout the region. We can plot this point in climate space and see that it receives 300 millimeters of rainfall and is 7 degrees C on average. Regions on the map most similar in climate to this point, those adjacent in climate space, are depicted in red. Note that even though a region may be geographically distant, those regions may still have high climate similarity to our point of interest. The most dissimilar regions are depicted as blue. In the context of plant materials, we might observe that a collection transferred from the origin of the white dot to blue regions on the map performs poorly relative to its home conditions. We can map climate similarity to multiple sources. Here we have performed this exercise for 3 sources, a Accession 1, Accession 2, and Accession 3, depicted as purple, green, and red points. Regions are colored depending on whether the climate is most similar to Accession 1, Accession 2, or Accession 3, in a process we refer to as assignment. Notice also that there is a color gradient within each assignment, various shades of purple, green, and red. This gradient depicts the range of climate similarity within an assignment, where darker, more saturated colors indicate greater similarity to the point to which it is assigned. Unsaturated regions indicate transition areas from one assignment to another. In the context of plant materials, each point may represent the provenance of an available seed source. In the assignment map, a suggested seed source for revegetation efforts throughout the region. We can also rank seed sources by their similarity to a region of interest, and use this information to select multiple sources rather than simply the closest match. In our example, we looked at only 2 aspects of climate, mean annual temperature and precipitation. However, climate similarity analyses may be conducted on any number of climate variables. Seed Selector conducts analyses on 7 climate variables that each contain unique and potentially ecologically relevant information on the North American continent. The selection process for these 7 variables is described in Doherty et al. 2017. These variables are a subset of the bioclim dataset, including mean annual temperature, diurnal range, temperature seasonality, temperature of wettest quarter, mean annual precipitation, precipitation seasonality, and the precipitation of warmest quarter. For more information on how these variables are derived, visit the address above or refer to Hijmans et al. 2005. The address to the Seed Selector app is linked above. When you navigate to Seed Selector, you get a welcome message stating that the app is initialized, a link to the underlying paper upon which the analyses are based, as well as instructions on how to run the app locally on your computer. We will get to this in a moment. However, the online version is limited in computational resources, so very large spatial analyses may not be appropriate for the online version. After clicking through the welcome message, you will see the graphical user interface of the app. In the center of the app, you will find an interactive map. To the left, you have controls to execute your analysis, and at the top of the screen are a few tabs for additional analysis outputs, a brief background on the app, and the instructions for running the app offline. Let's take a look at the controls working from the top down. The first items ask you to define your area of interest, the region in which you will conduct your analysis. At present, the extent is limited to North America, excluding larger bodies of water and a few islands. The default setting is to select your area of interest with two sliders that define the upper and lower bounds of latitude and longitude. If you click and drag an endpoint on a slider, you will notice that the extent of the green box on the interactive map changes to reflect the limits you select. You may also define your area of interest by uploading a spatial polygon. This could be a species range, ecoregion, jurisdictional boundary, or any map unit of interest in North America. You must provide a file compressed in the .zip format, which contains 4 components listed above of an Esri shapefile. Search for .zip file compression if you are unfamiliar with the process. The app accepts spatial polygons of any coordinate reference system. After you've uploaded your file, a polygon will render on the interactive map. Here we have uploaded a polygon of the Great Basin ecoregion, which we will use for an example analysis. After we have defined our area of interest, the next step is to upload a spreadsheet containing information about the accessions under consideration for a restoration project. This spreadsheet can be populated with conspecific data or a species mix depending on your objectives. This is an example of the data format that Seed Selector expects: an ID column, which accepts any ID label you choose, X and Y coordinates corresponding to the longitude and latitude in decimal degrees of a plant material's origin. Save the file as a .csv before uploading it to the app. Once you have uploaded your spreadsheet of accession data, the app will draw their points of origin on the map. The app assigns a unique color to each ID in the spreadsheet, which you can refer to in the accession legend key. You can also mouse over points to reveal their ID. Once both the area of interest and our accession data are uploaded, click the Match Seed to Climate button to begin the analysis. We will leave other settings as is for the moment. You will notice progress bar messages appear in the lower right-hand corner of the app, which indicate that the analysis is underway and which give an indication of the underlying methods as they are implemented. Analyses for ecoregions of this size usually complete within a minute. However, this is somewhat dependent on the level of traffic the app receives. Analyses of larger spatial extents or those with a greater number of accessions will take longer. The servers hosting the app may run out of memory and crash if the extent is very large. If you find that your analysis continues to crash after multiple attempts, this is an indication that you'll need to run them offline on a computer with greater resources. More on this in a moment. If you receive this message early on, you have selected a region for which the app has no climate data and cannot conduct an analysis. During the analysis, the areas within the Great Basin ecoregion were assigned to one of the 7 accessions that most closely resemble the climate properties of that accession. These assignments are depicted as a colored overlay. The coloration of an assignment matches its corresponding accession, and you may refer to the legend key to discern among assignments. If at any time you wish to turn off the overlay to reveal the underlying base map, we may do so by clicking the Overlays checkbox in the upper right-hand corner. If we zoom in on the accession gamma, we can see the similarity gradient within areas assigned to it. Darker shades of fuchsia indicate higher climate similarity to gamma. Lighter shades indicate lower similarity. The climate similarity legend key indicates the range of similarity values for analysis. A similarity value of 100 indicates identical climate conditions to the accession, and lower values indicate differing conditions. A value near 0 would indicate the lowest similarity possible within the region of our analysis. If we were to transfer gamma from its point of origin to areas of higher climate similarity, we might expect a greater chance of successful establishment as compared to areas of lower similarity. By clicking on the map at any point, we can extract climate information and a ranked list of the climate match of the accessions considered in the analysis. The numbers in the parentheses adjacent to the accession IDs are the climate similarity values of each accession to the click point. These pop-ups can help in assessing the magnitude of climate transfer from the home conditions of an accession to a potential restoration site. They can also help to develop a seed mix when choosing among many available options. If, for example, we had resources to purchase 3 accessions from a list of 10 available, we could refer to the climate similarity rankings to inform this decision. The climate rankings could also assist in provisioning for a genetically diverse population. Additionally, selecting multiple accessions could be a prudent strategy if conditions other than climate prove to be important. For example, plants might be locally adapted to a specific set of soil conditions, and the probability of matching these and other unknowns is higher if we select multiple accessions. We might also look at the climate gradients within an assignment to identify gaps in our coverage of climate space in a region. By making collections from lighter shaded areas in an assignment, we might fill these gaps. This process could be conducted iteratively, where we make a collection, add it to a second round of analyses, and then look for collections in newly calculated low coverage areas. Lastly, we might upload a single set of coordinates for a target site, and then conduct analyses across the region to identify climatically similar areas to search for collections. If we click on the Accession Climate Data tab at the top of the app, we can display the climate conditions of each accession's place of origin. Now let's click on the Within Assignment Distributions tab at the top of the app. After a loading period, you will see 3 box and whisker plots depicting the climatic distributions for our 7 assignments. This is a valuable tool for visualizing gaps in our sets of collections. If we look at the mean annual temperature plot, we can see that the majority of areas assigned to gamma are very close to 10 degrees C, quite similar to the home conditions of gamma. However, if we look at the temperature distributions of areas assigned to epsilon, we see that it varies by more than a degree from epsilon's home conditions. Even though home conditions of epsilon are the closest match among the accessions in this analysis, further collection within that assignment might be warranted to improve our climatic coverage of the area. To plot other climate variables, click the drop-down menus at the top of the tab. The next feature we will discuss in the tutorial is weighting of variables. When you click the Weight Variables button, the controls will expand with 7 additional sliders, each dictating the weight of the corresponding variables. Here we set all weights to 0 except for mean annual temperature and precipitation. Setting variable weights to 0 excludes them from the analyses. If we had instead decided to keep information from the other 5 variables, but reduce their importance rather than exclude them entirely, we might set their values somewhere between 0 and 1. Setting variables to 0.5 would equate to half the importance of an unweighted variable, 0.25 a quarter, and 0.1 a tenth. When is it appropriate to weight variables? You may have prior knowledge of a species' response to climate transfer derived from a common garden or reciprocal transplant study and wish to improve chances of successful establishment based on this information. If you possess empirical data such as this, you might first model plant performance as a function of climate transfer across the 7 seed selector variables, using an algorithm such as random forest, maxent, or boosted regression trees. The next step would be to extract the variable importance metrics from the model, and then identify the maximum importance value, and finally divide all importance values by the maximum. These relativized importance metrics could then be provided as input weightings to seed selector. After running an analysis, you can download the relevant outputs to your computer to explore in your own mapping software. To reproduce the aesthetics of the interactive map, set the SimVal TIFF to a black-to-white gradient, then overlay the Accession assignment TIFF, setting it to 50% transparency, or as desired. The assignment valkey.csv documents the raster value associated with an Accession ID in the assignment TIFF. Assignment coordinates and climate information are found in the Accession data CSV. Box plot figures for the assignment distributions may also be found in the PDF file. We now provide the option to run Seed Selector on your own computer for larger analyses. Instructions for this process may be found in the Run Offline tab. Installation requires some knowledge of the R programming language, in addition to a few actions in the command line terminal. We will update this tab in the future if we can develop an alternative, more accessible solution. If you are a programmer or scientist who would like to explore the source code of the app, it is available in a GitHub repository linked in this tab. Please note that while you can leverage the power of your personal computer to conduct these analyses, you still require an internet connection to display the base maps. There are numerous potential directions we might take this app, one of which is to incorporate future climate data, which could be used to assess the future utility of accessions and plan for assisted migrations. There are also now globally available soil grid data, which could be added to study the importance of soil chemical and physical properties. It would also be beneficial to extend the coverage of the analyses globally and make them quicker. If users encounter any bugs, have questions, or wish to collaborate in future efforts, please click the Contact Info button in the app and send an email to the address provided. Seed Selector is primarily a tool for picking among available accessions for use in restoration projects. We also serve a complementary app, Climpart, linked above, that provides spatially explicit guidance for seed collection. I'd like to thank the U.S. Geological Survey and Colorado Plateau Native Plant Program for supporting the research and development that went into these products. I'd also like to thank the Botanic Gardens Conservation International for assistance with developing this learning module.